Welcome to solving protein synthesis problems. This is probably the most challenging aspect of this section of the unit. And yeah, if you find it that challenging, I highly encourage you to make sure you watch the previous podcast, watch some of the crash course videos on DNA and RNA, watch the protein synthesis ones, because that's really what this is all about, is how that nucleus with its code is actually translated into the specific proteins that you need. So this worksheet we'll come back to in a second, but yeah, I just wanted to say a couple more things about this molecule, and then I'll show you a few examples of how to do the worksheet, leave you with a few of the problems, and hopefully you will master it and feel like you are now yeah, a budding molecular geneticist. So go for it. The importance, again, is to realize that the code in DNA actually codes for the making of protein. So we're calling this protein synthesis, the making of protein. Synthesis simply means to build up, and protein is important. As you can see in this diagram, so many areas of your body are made of protein. You are protein. Hair and nails contain keratin, fingernails. Blood contains hemoglobin, a very important protein that actually is responsible for transporting oxygen in the blood. So many other things in the blood, the antibodies that fight disease, enzymes in your digestive tract, hormones, muscles. Protein is important, and so every time your body needs these proteins, it's absolutely critical that the recipe for them has been perfected, and that recipe is carried out. And how does that happen? Protein synthesis. So just a few characters that actually are instrumental in this process. When you look at these dudes up at the top, what you're looking at is a picture of two gentlemen by the name of Watson and Crick. Watson and Crick actually won the Nobel Peace Prize. Well, we wouldn't call it the Peace Prize, would we? We'd call it the Nobel Prize in Science for discovering the structure of this double helix. This is the double helix DNA, and that structure was very much based on research where they actually knew what the nucleic acid DNA was made up of, in particular 4 bases that contained nitrogen. They were adenine, thymine, guanine, and cytosine. Those bases they actually analyzed and began to come up with some rather interesting data. And if you want to win a Nobel Prize of any sort, you might want to look at that data and just see if you can come to the same conclusion that they came to. What I see is when I look at the ratio of adenine to thymine, or A to T, they seem to be equal, or within experimental error, are pretty close to being equal. The same thing is happening if you look at guanine and cytosine. Regardless of what living thing we have on this planet, always G equals C and A equals T. So if that is the case, maybe, just maybe, the structure would somehow point to the fact that A must actually be attached. Like in this diagram, you see A attached to or bonded to T, G attached to or bonded to C. And that is, of course, what turned out to be the case. The base adenine is bonded by a hydrogen bond to another nitrogenous base, thymine, A always with T, and guanine always with cytosine. Young lady by the name of Rosalind Franklin. Rosalind Franklin. You may have heard of the Franklin Institute, named after this young lady. She probably should have been given a whole lot more credit for the discovery, because she was actually the one doing the X-ray crystallography, looking at X-ray pictures of the molecule and realizing that if in fact the A actually paired with the T and the G with C. She unfortunately was a woman in an era in science where women weren't given a whole lot of credit for scientific research, and she also happened to be young. She also happened to be Jewish. a whole lot of strikes against her. And the saddest part of the tale, of course, is she actually died very young of cancer as well. So posthumously, we recognize her as being very instrumental and probably should have been part of that Nobel Prize. I'm pointing out a couple of books to you here, The Violinist's Thumb and The Language of God. Fascinating books that are available even in our school library on this whole area of molecular genetics and genomics. The Language of God was written by Francis Collins. Francis Collins is a very interesting character, also part of the American team. In fact, he led the American team in the search for the Human Genome Project. And let's just put it this way. Either one of those books, if you were to read them, would be so much more valuable to you than any one of the silly little assignments that I may conjure up for you to get the basics of this process. So if you're interested in reading those, do feel free to take them out of the library or get yourself a copy, and we'll just have a little discussion about what you learned, and we'll call it a done deal as far as what you need to know about DNA and molecular genetics. So what I think we should do is actually move through the last little bit of information, and then I'll show you the worksheet. I must confess my own interest in molecular genetics probably was given a kick in the backside when I pulled out of Vancouver Province a few years ago, and the cover of the Vancouver Province paper said, "Revealed the secrets of who we are." And what it was, of course, was the culmination of the Human Genome Project, but it had a little subtext to it. The most amazing story of human life, how we got here, where we're going, is revealed by the map of our genes. And got me thinking that a lot of the attributes we tend to think of in children, or in your case, future children, actually are genetically controlled. And by knowing the actual genes on the specific chromosomes, we now seem to have this window into very much our future as well as our children's future. So I got thinking hysterically absurd ideas about, hey, wouldn't it be nice to actually have a 7'4" daughter that could play a middle in volleyball, or have incredible skills on the piano or other intellectual skills, because we now know where certain genes are that code for these. Probably more realistic is just the realization that for our children and ourselves, we would like to avoid things like cancer and dementia and a whole lot of other nasty diseases. So yeah, the Human Genome Project is at least the first step in coming to a far better understanding of what those genes code for, and then ultimately perhaps having some control over them. So in a sense, I look at DNA as essentially a barcode. A barcode, you go through a check stand in your favorite supermarket, cashier goes beep, and the computer immediately knows that you've just bought a certain product and immediately orders a new one and replaces certain orders. Just a ton of information contained in the sequence of these bars. But if we think a barcode contains information, the information coded in your DNA molecule is just so much more significant. The point is that when that code goes wrong, we have a mutation, and mutations can lead to a whole lot of disorders, and the ones we tend to think of are the ones that turn up in circus sideshows, the bearded lady and that sort of thing. But the truth of the matter is, if you take a chromosome like chromosome 9, got a certain cancer at one location on that chromosome. By the way, the fact that these are shown as single chromatids tells you that these are chromosomes that haven't undergone replication yet. Prior to cell division, the cell is in a stage where it's simply carrying out its function and hasn't entered the synthesis stage where replication and protein synthesis is going to follow. So each chromosome now, following the Human Genome Project, has a map that shows us that certain types of diabetes, breast cancer, diseases like phenylketonuria, the inability to synthesize, not synthesize, but process the amino acid phenylalanine, a lot of diseases that are going to affect lifestyle are actually controlled by the sequence of genes, a code, right? So this has huge consequences socially, economically. If this information is actually recorded someplace, and you can basically find out your own genetic code by sending a small saliva sample to any number of labs around the world now. The question is if you really want to know that information. Once you know that you're likely to come down with Alzheimer's, but there's no cure for Alzheimer's, does that really improve the quality of your life? The big question again too is, where is God in all this, and are we really living in a deterministic society where we really have little control over our actions, our children's actions, who they are, who we are? I choose to think not, but the fact remains that if this information is contained in a repository, say in your schools, your universities, your future employers, is it likely that you as an employer are going to hire somebody that has potential, Huntington's chorea or cystic fibrosis, or you see long-term that they're likely to develop heart conditions, or all of these things are going to cost your company money. So what we have is deliberate choices by corporations to not hire certain people, and you can imagine the consequences in society of that sort of thing. But just a brief outline of what this is all about. Finish up with the protein synthesis package that you need to turn in. But again, I encourage you to you may think of going through those videos as a waste of your time. The fact of the matter is it's actually the best possible use of your time. So we're talking about the Human Genome Project. Obviously, we're talking about genes. So what the heck is a gene? The concept of a gene, everything that involves energy in your body, as you probably know by now, is controlled by a huge number of enzymes. We've done a chapter on enzymes already, and you know that there are thousands of them in each cell, that without them, reactions don't go from substrate to product. So it's the genes that actually code for the production of those enzymes. Many of these enzymes are, of course, proteins, and that's what DNA codes for, is protein. These genes can be regulated, and again, that's controlled, again, by genes, other genes. The other factors can regulate the activity of enzymes after production. The point is, the big picture here is that you start off with a DNA code, a double helix, represented by the sequence of the bases. DNA, of course, consists of rungs of a ladder that are held together by hydrogen bonds. These rungs of the ladder are basically nitrogenous bases. The nitrogenous base thymine, always paired with adenine, guanine, always paired with cytosine. Along the sides of the ladder, you have a sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. At some point or other, that sequence of bases on the DNA has to be read and transcribed into a different nucleic acid, messenger RNA. So mRNA stands for messenger RNA. Deoxyribonucleic acid has the master code. It is the recipe essentially for life and every protein and enzyme in your body. But at some point or other, when your cell needs it, a section of that DNA is going to be opened and read. You may be familiar with the term scribe. To scribe is to write. So what transcription is all about is taking that code and putting it in a form that can actually leave the nucleus. DNA is in the nucleus, mRNA leaves the nucleus and eventually attaches to a ribosome, where the last stage of protein synthesis takes place, translation. The whole thing is about making protein, right? So our code that is transcribed now has to be translated into protein. And that's where the rubber hits the road. That's where the metabolism really comes in. So more will be said about this process, but for now realize that before this ever takes place, DNA makes a copy of itself. You're not going to mess with the master code. You want to have a backup copy. So the actual copy that is used for transcription, you undergo replication first. So the three processes: replication of the DNA, then transcription, finally translation. This is simply a fleshed out version of the previous diagram. Again, just emphasizing that it's genes that code for the production of proteins, which we mentioned are enzymes usually. So you start with the DNA code. At some point or other, that code has to be transcribed into messenger RNA so it can leave the nucleus and actually attach to a ribosome where the protein is actually made. So if you have a thymine nucleotide on the DNA, that means the complementary base is going to be on the messenger RNA. T always pairs with A, so mRNA is going to have an A there in that position. If you have a G in the position on the template strand on DNA, then you're going to have a cytosine, or C, on the messenger RNA. You may notice on the messenger RNA there is no thymine. So no T. And the way I remember that is the word not. So no thymine in messenger RNA. So everywhere you have an adenine on the DNA molecule, you're now going to have adenine pairing with another nitrogenous base called uracil. U stands for uracil. And you're not going to find uracil in DNA, but you are going to find it instead of thymine in messenger RNA. And again, that messenger RNA leaves the nucleus, moves to the cytoplasm where it attaches to a ribosome. There's all these amino acids attached to what are called tRNA, or transfer RNA molecules. They will attach to the 3 bases called codons on the messenger RNA and make a protein. And those proteins could be structural, they could be regulatory, contractile, things like antibodies, transport proteins like the ones in your cell membrane, or enzymes, catalytic proteins. So, we have said that genes control protein synthesis. Where do you find the genes? Genes are found on the chromosomes in the nucleus. So, again, chromosomes normally are not nearly as pretty as these sweet little artistic versions here. They're more likely like this. When they're not paired, they exist as single chromatids. The banding patterns really are where you're going to find the genes. We're not going to go into a whole lot of detail here because a lot of this you may already be familiar with, but DNA is usually not found on its own. Before cell division, it's found loosely as chromatin. Just before cell division starts, however, it condenses and it actually attaches to what are called histone proteins and forms these coils. And again, the purpose of that, it's believed, is to basically save space within a nucleus. A nucleus is not that big, and you have an entirely massive tome of information that has to be somehow stored within that. So all of the packaging and compression that can possibly go into it is applied at that point. So a replicated chromosome is double-stranded like this. The non-replicated chromosome simply exists as chromatin. When you look at a cell, even a human cell undergoing cell division, mitosis, you are going to see a whole lot of chromosomes that aren't nicely paired like these. So in order to look and study chromosomes, you have to do what's called karyotyping. Karyotypes display the chromosome contents of a cell organized according to their number, size, and type. So you eventually pair chromosome 1, pair chromosome 2 based on their size, and you're going to notice that they have exact copies at the same locations of the gene. One comes from your mommy, one comes from your daddy. Normal human body cells, these are called somatic cells, have a karyotype with 46 chromosomes, existing as 23 pairs. Of those 23 pairs, 22 are called autosomes, and then depending on whether you are a female or a male, you are going to have 1 pair of sex chromosomes. So in the case of this guy is a guy, he's a male, he's got this little broken chromosome called a Y chromosome, and there's a whole lot of interesting information about what in the world genes are on that and why it looks so small and all the rest. But read, the violin book, and you'll find out a whole lot more about all of that interesting information. Just to quickly look at a human female karyotype, though, every cell in a normal human cell except egg cells. And of course, the reason for that is egg cells undergo meiosis, so you have half the chromosome number. You have 44 autosomes and 2 sex chromosomes. So these are the 2 X chromosomes in the female. And in the case of the male, we have, again, 44 autosomes, 2 sex chromosomes, 1 X chromosome. This is, of course, why there's a lot of genetic diseases that are only shown up in males. Sex-linked diseases like color blindness, where it's passed from the mother to the male son, is going to is going to actually express itself because the male only has 1 X chromosome that he's got from his mother, right? The other one is a Y. So, back to DNA. DNA is made up of nucleotides. The building blocks of nucleic acids are nucleotides. And we're talking about nucleotides whether we're talking about DNA or RNA. All nucleotides have a sugar. In the case of DNA, it's deoxyribose. In the case of RNA, it's ribose. They all have a phosphate group, which of course is actually this PO4. And they all will have one of 4 different types of base. Adenine, cytosine, guanine, thymine. A closer look at a nucleotide. Again, you see the phosphate, the sugar, attached to the purine base guanine. It's not a big thing in this course to distinguish pyrimidine bases from the purine bases. The way I tell the difference is you'll notice that pyrimidines have the letter Y in it, cytosine, thymine, right? So just like pyrimidine, Y. And the purines, of course, like adenine and guanine, don't have a Y. You may wonder what these bonds are here connecting the nitrogenous bases. Those are hydrogen bonds. Those are relatively weak bonds because during replication, during transcription, they actually have to be unzipped and reattached, and they can reform rather easily. The genetic code is actually based on the sequence of amino acids. So the code is read in a sequence of 3 bases. They're simply called triplets on DNA. But when you have those triplets transcribed into messenger RNA, we call them codons. So you will get something like ACU or GCC. All of those are considered codons, and you will always be given a chart in order to translate those codons. And the only way you can translate the codons is by having amino acids that actually match these. And they, of course, are found on the transfer RNA molecule, which contains something called an anticodon. More to be said later. You see why you want to watch the animations. You see why you want to watch those Campbell activities. You need to see this in action. So each triplet codes for 1 amino acid, but more than 1 triplet may encode some amino acids. The code is said to be degenerate or redundant because you have more than 1 possible code, which is actually a good thing. If you look at your chart of codons, you'll notice that if you had a mutation where the last base was actually changed, often you'll still get the same amino acid. Then there are actually some codons which don't code for amino acids. They are codons that tell the cell to actually start, which is AUG, and then there are codons which actually stop the protein so you don't keep adding amino acids. So an amino acid added to an amino acid, joined to amino acid, of course, is a polypeptide. At some point or other, you have to stop the addition of the amino acids, or you're going to end up with the wrong protein. So below is a messenger RNA strand coding for 6 amino acids, 1, 2, 3, 4, 5, 6, and AUG would be the start codon. UAA or methionine would be the stop codon. Before you ever make a copy, before you ever actually start protein synthesis and begin to transcribe a sequence of bases on DNA, you want to make sure that you have a backup copy. So DNA always makes a copy of itself. It's called replication. This takes place in the nucleus. DNA is replicated to produce an exact copy of a chromosome in preparation for cell division. The first step requires that the coiled DNA is allowed to uncoil by creating a swivel point. This is a replication fork. The swivel point basically is created by enzymes, and there's a whole lot of enzymes that quite honestly you're not really terribly responsible for. I'll show you some of them here, but yeah, don't start hyperventilating, because the truth of the matter is you only need to understand that these steps in each of these processes is controlled by enzymes, but only very few of them would you be considered responsible for. So I'm not going to go into a whole lot of detail on this replication. You can look at the diagram here yourself. You can read as well as I can. It's much better to see this process of replication, the process of transcription and translation all in animation, rather than simply relying on a two-dimensional diagram. The point is, when replication ends, you now have two new strands of DNA which are identical. So everywhere you had a guanine, you're going to have a cytosine. Everywhere you had a cytosine, you're going to have a guanine. The reason this happens, of course, is you have a whole lot of free-floating nucleotides, one with thymine attached, one with cytosine attached, adenine, etc., floating in the nucleoplasm of the nucleus. If you get nothing else out of this section here, just make sure you know the base pairing rules, that adenine always pairs with thymine, guanine always pairs with cytosine. So let's get back to this solving protein synthesis problems. That's probably what brought you here in the first place. I'd encourage you to read this first paragraph. Hey, you know what? I'm just going to read it to you. Kindly follow along, and it's a good thing for your brain to actually hear some of this vocabulary, and it eventually puts it all together. So you are made of proteins, structural proteins like the ones found in hair, skin, nails, contractile proteins like the ones found in muscles, catalytic proteins, which are enzymes which control the rate of thousands of chemical reactions going on in your body cells at any time. You have transport proteins like hemoglobin that transport oxygen in your blood, and many, many more. These proteins are made up of building blocks called amino acids. Don't be afraid of the word amino acids. Yeah, they're just the building blocks that make up all protein. They have funky names such as leucine, proline, serine, etc. The type of protein is determined by the order of these amino acids. So again, it's the order of the amino acids that make up the protein that make the protein what it is. In other words, there's a recipe or code for each of these proteins. That code is found in the nucleus of each cell of the body in the form of the nucleic acid deoxyribonucleic acid, otherwise known and loved as DNA. DNA is so very important that before it gets to work making the proteins needed at any time in the body, it first makes a backup copy of itself. This making a backup copy is called replication. So, let's just say the following is the base sequence of one strand of a DNA molecule. Again, DNA is a double helix, so one strand is just the order of the bases down one half of that double helix. And again, get familiar with the symbols for the various nitrogenous bases in DNA. Adenine we will abbreviate capital A, thymine capital T, guanine capital G, and cytosine capital G. So the first question, give the base sequence of the complementary DNA strand. So in other words, what are the bases that are joined to adenine? So as you know, adenine is always, always, always paired with thymine. So we have another adenine, so there's going to be another thymine on the other side. Then we have a thymine, so adenine is going to be on the complementary side. So TTA will be attached to AAT on the one strand. Where you see GCC, we'll have CGG, and where you have the next three, sometimes it's easy to get mixed up here, so it's a good idea to go in threes. We've done AAT, GCC. The next three are AGT, which is going to be thymine for the A, and then we're going to have cytosine and adenine. And if we just continue going down the strand, cytosine, cytosine, adenine, adenine, guanine, cytosine, and then the last three bases on the DNA, CAC, will be attached to GTG. Right. So, next question. Draw this DNA molecule down the right side of the page. And again, you don't have to do the entire strand, but just kind of give me the idea that you know what you're doing here. DNA, as you know, is a double helix. Down the sides of the ladder, you've got this sugar phosphate, sugar phosphate, sugar phosphate. So the sugar that we're talking about here is deoxyribose, and it's attached to a phosphate. So that's a P, joined to another deoxyribose, joined to a phosphate, joined to another deoxyribose, joined to a phosphate. You get the idea. So the same thing is going to be happening down the other side. Another sugar, deoxyribose, joined to another phosphate, joined to another sugar, the deoxyribose, joined to phosphate, joined to the deoxyribose, joined to phosphate. Now the question is the bases. Maybe we should switch color here just so we know. Yeah, just keep track of which color we're dealing with here. Let's go for the base that is attached to deoxyribose. Deoxyribose, again, is going to be attached to, notice that it's A. So deoxyribose is A, and adenine is attached to, okay, that's an A, honest, is attached to thymine on the other side. Let's get to the next sugar. The next sugar is attached to A again on the original strand, and then on the complementary strand, it's attached to T. So this basically is this strand, and this one over here is the complementary strand. So the next base is going to be thymine, and it's attached, of course, to adenine. Then it's just a matter of attaching the various bonds to it. So deoxyribose is actually attached to adenine, thymine is attached to the sugar, and then you will actually have the two bases attached by these weak bonds called hydrogen bonds, which by convention we just go dot, dot, dot. And for some reason or other my dot, dot, dot is not turning up, dot, dot, dot. That basically will take care of. Maybe you should try to do about another 3 rungs of the ladder. But notice the base pairing, A with T, A with T, T with A. It's basically A with T, A with T, T with A. Number 3. Now this is a paragraph you might need to read over a couple of times. Let's just say that this section of DNA above is a gene that codes for the production of a protein. In order for the code to be used, it has to leave the nucleus. But DNA can't leave the nucleus. What to do? DNA has to send a messenger that carries the code to the ribosomes, those little protein factories in the cytoplasm of the cell. The messenger is that other nucleic acid called messenger RNA. That's what the M stands for. The process of mRNA forming is called transcription, or the process, the verb is transcribing the original DNA into a copy. So be careful here. RNA includes the bases adenine, guanine, and cytosine, just like DNA, but there's no thymine. Think of the word not, no thymine. In place of thymine, RNA has a different base called uracil. So G still pairs with C, but now A pairs with U. So the question here is down here, give the base sequence of the strand of mRNA that would be formed from the original DNA strand above. So the original DNA strand is this one right here, and now we're talking about RNA. So RNA, everywhere there's an A, there can't be a thymine, there's going to be a uracil. Everywhere there's a, the next one is A again, so there's another uracil. Then the next one is going to be T. T bonds with A. Look back at guanine. Guanine pairs with cytosine. Guanine, guanine, uracil, cytosine, adenine, and if you just finish off the strand, you should get CCA. So, just in case you were missing that strand of messenger RNA that was transcribed from the original DNA, we're now on the next page. Right at the top of the page, it might be useful for you to actually write down UUA. So let's get a pen going here. Yeah, that should do it. UUA. And then the next three would be CGG, followed by UCA. That'll never do. That's not good. UCA, and then CCA, CCA, AGC, and GUG. So the question is, we now have a copy of the code for a protein that can leave the nucleus through a nuclear pore and attach to the ribosome. Each 3 letters of the mRNA read from left to right make up what are called codons. Yes, these codons tell another RNA in the cytoplasm called tRNA or transfer RNA to bring a specific amino acid to the ribosome where they can be joined together in a long chain called a protein. Use the table of RNA codons to determine which amino acids this mRNA molecule codes for. So this is a table of mRNA codons. You can't use this for DNA, you can only use it for RNA. And you're given this chart if you ever need it on a test. You're not supposed to memorize it. As long as you've got these codons, you can just look at UUA, find UUA in the chart, and you should be able to find, let's find UUA on the chart. Okay, I'm looking, are you looking? Yeah. UUA, down here, leucine. So we know the first amino acid in this protein is leucine. That's an N. And it's joined to another amino acid, CGG. CGG is going to be arginine. Right there, CGG. Whoops. CGG. Notice that there's multiple copies of these codons. CGG right here. In either case, it's going to be the amino acid arginine. I got to work on my printing. Arginine. If anybody knows the secret to making a Surface Pen actually work, I would be really thrilled to, hear from you. So let's move that back just a tad here so you can actually see it. The next codon we need to look at in order is UCA. Find UCA on the chart. Let me see. UCA appears to be serine. Serine, that's an E. Honest, it's an E. Serine. And then the next one is going to be CCA. This one right here, that codon. If you look at CCA in the chart, it's proline. And then AGC, the next codon. Where is AGC? It's going to be serine. Serine. And then finally, the last one, GUG. If you look on the chart, that is going to be down here, valine. So these are all amino acids. The more of them you get, you have what's called a small protein or a peptide. A polypeptide over 50 amino acids is called a protein. So number 5, it's now your turn. We're giving you another sequence of DNA, just like on the first page. If you follow what we did on the first page, transcribe that into messenger RNA, use the codons, you should be able to move on finally to that last page, which is going to really bless you because you're going to have to think about it. So, especially a couple of these questions at the end, I'd encourage you to give the bonus question a try. And if you have any problems, talk to me in class because, yeah, you can make this happen.